Delighted we've got this opportunity to talk tonight. There's quite a bit of ground we can cover. We don't have an infinite amount of time, despite your love of stories. So we'll try to keep this more or less focused. And I thought a good place to start would be for you to tell us a little bit about your education, high school, college,
Sure.
and up to the point where you made this decision that you were going to go to medical school as opposed to getting a PhD.
Sure.
So.
Well, I should tell you, in those days, you listened to your father. This goes back a ways. And he was a former United States Marine. And his view of life is that if you're a naval officer, you're one step below God. So he got an appointment from a congressman in St. Albans to send me to the Naval Academy, which meant that I went to Cooper Union School of Engineering, because that's what you do to prepare to go to the Naval Academy. And fortunately, I flunked the physical examination. I was too myopic, and I had a transient adolescent hypertension, actually, as well. That helped me, saved me for that. So Cooper Union, it's an elite institution. You had to pay an examination, take an examination, and I passed it. And there was a small class of about 40, 45 kids. And I quickly learned that I was the second from the bottom of the class. When I flunked the examination, I said, I'm out of here. I wanted to do biology. So before I could leave...
I'm not quite sure I understand that connection.
Yeah. Well, I didn't.
We could
I wasn't
expand
very
on
good at
that
civil
later.
Engineering. Yeah, Office of the Navy. So anyway, there was a mandatory interview by the school psychologist before you could leave. And he said, George, I voted against your admission. He said, you'd never make it here. You played jazz trumpet. Yeah. He said, you were on the track team in high school, and you go out with girls. Yeah. You'll never make it here. So anyway, I went to CCNY, and I was interested, among other things, I was quite interested in social psychology in those days. I thought that was the big problem in the world. And still is a big problem in many ways. So then I got into the Navy by passing a special examination where they waived the physical requirements. It was called the Captain Eddie's Electronic Training Program. They didn't know, they had these sonar and radar, nobody knew how to fix them. So if you could do powers of 10 real fast and know about parallel and serial circuits, they would take you in as a seaman first class. So that's what I did, and got out of the Navy. Was with a guys who used to fly to Alaska, and I was just a wild kid trying to try something new. And I went to Alaska. I worked on Alaskan Railroad as the joint spiker, which is a very prestigious job, and saved about $1,000 in 1946, and went to the University of Alaska and applied to medical school here, and they didn't believe my grades. I had like straight A's, so they forced me to come here for the last year, which I did, and I got straight A's except for, I was a musician, except for music appreciation. I got a C in music appreciation. I didn't pay any attention to it. But anyway, pre-medical education is one piece of advice for, well, the students here are too advanced, but I think it was a terrible education, frankly. It's a smattering of ignorance. I had physical chemistry, and then I realized across the hall there was real physical chemistry. Mine was physical chemistry for pre-medical students. No. So they didn't pay much attention to us. So that was a problem. Going to medical school, I had always had some interest in behavioral science and neuroscience. I had, my youngest brother had Down syndrome, so there was always interest in genetics. It was not an advanced maternal age situation, as it often is. And I had a sister, an older sister, who had meningitis before the days of antibiotics, and she was deaf at the age of 4. So that was a background for being interested in medicine. And when I got to medical school, I guess it took about 2 lectures in psychiatry to realize that this was in the Stone Ages. I got out of there fast. Yeah. And what else happened in those days in medical school? Well, I was influenced by H. Stanley Bennett.
Yeah, I was wondering if there were particular either teachers you had or formative influences then.
Yeah, so he was the
Stanley,
chair.
And he was here.
He was here, the chair of biological. It was called anatomy in those days, and he was a pioneering electron microscopist in nature of the unit membrane. And he brought Linus Pauling in, and Linus Pauling had been to a dinner with Dr. Diamond in Boston, a hematologist, and sitting next to him, he described this business of sickle cell anemia. And Pauling said, well, it must be an abnormal hemoglobin.
Yeah.
So he went back to California and showed it was an abnormal hemoglobin. He talked to us about that. He talked about basically the birth of molecular genetics, and I thought that was really exciting, even though my goal at that time was to be the first pathologist in Alaska. They had no pathologists. I could have made a fortune if I stuck to that. It didn't happen. And then at the clinical level, Belding Scribner, he's the guy who designed the first shunt, arteriovenous shunt for renal dialysis.
Right.
And he taught me a lot about fluid electrolyte balance, and I got an honors thesis with him on resistance to mercurial diuretics in congestive heart failure. So that was part of my influence. The other guy was Stuart Lippincott. If you go to our department of pathology, you see all the chairs.
Right.
They left out the first chair. Student Lippincott. There's no picture of him. And he was chair for the first few years. And
but this was prior to the medical school.
This was prior to medical school.
Formally.
No, this was in medical school. Yeah.
Okay.
So I'm jumping ahead.
All right.
I got into medical school here in Seattle, and he was talking about what to do in residency. I still wanted to do pathology. And he convinced me to go to Montreal General Hospital and McGill, and because there was a wonderful pathologist, G. Lyman Duff, who was a pioneer in the study of atherogenesis, and atherosclerosis was a big thing in pathology, still is the major cause of death in developed societies, coronary artery atherosclerosis, and also strokes. So I arrived, and the poor guy had just been diagnosed as having metastases from bronchogenic carcinoma. He was a heavy smoker. And also, I would starve to death, I think, in Montreal at the wages I got. So I went to the University of Chicago. And in those days, it was wonderful. The University of Chicago, I got in via a telephone call from the dean, Dean Turner. That's all I did. He just called him up. He said, George is a good guy. So so the chairman, Paul Cannon, asked me to come to Chicago. And that your home education.
Where did the motivation for pathology come from? I mean, you mentioned a couple of family instances
that was
here
it.
that might have been.
Well, I came to the conclusion that was the best deal because you got paid to learn. You got the answers. You got
as opposed to paid to do?
Yeah, right. Whereas, in internal medicine, you're sure guessing, you have a differential diagnosis. We saw what was happening. I thought that was great. It paid to learn. I still think it's great. So, anyway, what happened? Should I continue? Or you want to ask me a different question?
Well, we're in Chicago.
We're in Chicago now,
and, and there was a connection made at that point, I guess, with Earl Bendit, who was there as a faculty member.
Correct.
And when Earl decided to come here to found the Department of Pathology, I guess the formal Department of Pathology in the School of Medicine, he extended the to you to come and join him.
And my wife, Julie, was thrilled to come back home. And actually,
she had grown up here.
She had grown up here in Bothell. And so, anyway, my mentor there was Eleanor Humphreys, whom you may remember, superb surgical pathologist. I was a hotshot surgical pathologist, which what Earl Bendit wanted me to do, that diagnostic stuff. They needed that here. So, I was his first appointment. And I came, and one of the things he asked me to do was since I was interested in genetics, and I tried in Chicago, I tried to produce Wilson's disease by giving copper to mice. Didn't work. So he said, well, George, why don't you just do the 3-week segment in the introductory pathology course in genetics? So I immediately went to these two guys, Arno Matosky and Stan Gartler. I said, could you help me?
Yeah.
And we've been close colleagues ever since, and it was one of the best things I've ever done. And I try to wean myself away from diagnostic pathology. And I remember the first couple of years, the residents would crawl up several stairs to wait in my front of my office to show me the slides.
Yeah.
But after a while, they stopped showing me slides. So that was good.
At some point in there, though, you made the transition to doing more lab work. And what I mean,
I got to tell you one episode, though.
All right.
Anecdote. One of the things you may remember I did in that course was introduce laboratory exercises. I was interested in those days in ceruloplasmin because it's very low in this disease, Wilson's disease, which we now know it's due to a copper transport mutation. So the ceruloplasmin doesn't get its copper, and it's very, very low, and it's a diagnostic. So I had the students do assays for measure ceruloplasmin, and then I learned that the girls who had high levels of ceruloplasmin were on birth control pills. So that was a bad thing to do, so I stopped that.
That exercise was deleted in subsequent years.
Deleted that. The other thing we did, we just had karyotypes in those days. Phytohemagglutinin was. And that's an interesting story of how that came about. Anyway, one of the students was mosaic for Turner syndrome. So that was a problem. So I stopped doing those laboratory exercises. Yeah. So anyway, that's how it got started.
So the research, though, at some point took off. And what were the things that you were working on when you first started and built the lab and started to do lab work?
Right. The first thing I did, I was interested in things that related to copper, and I wondered if there are other prosthetic groups, and I looked at vanadium. I thought, that's interesting. And I published a paper in Nature showing that the non-enzymatic catalysis of oxidation of a bunch of biogenic amines. I was still interested in 5-hydroxytryptamine, serotonin, dopamine, noradrenaline, adrenaline, and vanishingly small amounts of this stuff would turn it black. So I was really impressed. And I started reading the literature, and some of it had to do with fungi that maybe required trace amounts of vanadium. And by reading this, I got to read about Aspergillus nigilans and a guy by the name of Guido Pontecorvo, who had discovered the parasexual cycle in Aspergillus nigilans and suggested, hey, human beings have the first step in the parasexual cycle. They're heterozygous at a bunch of loci. And there, for Aspergillus, you did haploidization from diploids, and they had mitotic recombination, mitotic crossing over, and Ponte, we called him Ponte, his nickname. You could show the linear order of the genes are the same as meiotic stuff. I was really excited about that, and fortunately, Earl was very friendly with Herschel Roman. Anne is here. It's her dad, who is the first chair of the department, and so is Stan. And I got to know them socially as well as my contacts in genetics, and it was a wonderful experience. And I think the social aspects are really important in science. They treated me wonderfully, and I invited me to dinners, and so did Arno and Gretel. Also was a very valuable experience as a young, very naive. I'm still naive in so much of genetics, but that was wonderful to accept me that way. And so I told Herschel about this, and Herschel wrote a letter to Pontecorvo, and Earl let me go on a sabbatical, even though I started in 1957. I went on a sabbatical in 1961, and it was a fantastically good year.
And this was in Glasgow?
This was in Glasgow, University of Glasgow. My daughter came back with this wonderful Scottish accent as a 4-year-old, and she unfortunately lost it in a year. So anyway, this was 1961, and I was doing serial subcloning and replicate plating, and it was a disaster because we now know
with Aspergillus.
No, this was human cell.
Oh, these were human cells at this point. You'd made the transition.
Yeah, because and I told Ponte, this is ridiculous. You can't do genetic analysis. This stuff, it stops growing. So he said, nonsense. He said, you guys get the right medium. You don't have the right medium. So we kept struggling, and when what we did, the 2 or 3 of us who were doing somatic cell genetics with human cells, we would sneak back to the lab at night and work on Aspergillus. So, but it turned out that I finally got interested in asking the question, does this really have something to do with aging? Because Leonard Hayflick And Paul Moorhead came out with the notion, with that famous 1961 paper showing that this was a general effect and that it may be aging at the cellular level. Then I remembered work that I did with Arno, and with Arno's outstanding postdoctoral fellow, who was interested in Werner syndrome and asked me to do the pathology. And so we did a review paper on...
So it was Charlie Epstein.
Charlie Epstein, of course, did a review paper on it. And I said, well, maybe if this is related to aging, since they had a phenotype that suggested of accelerated aging. Later, I called these phenotypes segmental progeroid syndrome. I like oid because it means like. Pathologists love that, oid, because it's like something, but it doesn't mean it's exactly like something. And actually,
it's a weasel word, I think.
It's a weasel. Rudolf Virchow, he invented the term amyloid because he did an iodine test for starch in these waxy livers with a different kind of amyloid, and he was smart because it turns out it's a protein. But coming down with the protein is heparan sulfate proteoglycans. So he was, anyway, I call these segmental.
You're going to learn more than you want to tonight.
Yeah. So
George is an education.
Yeah. So at any rate, where were we? I lost. Yeah. We were an amyloid. Okay.
No. You and Charlie and Arno.
Oh yeah,
and Amelia Schultz had seen these Werner patients. And,
I'm on track again.
And you were going to try to grow cells because you thought there might be a phenotype related to the organismal phenotype.
And you come into this story soon too.
Much later.
So yeah. So anyway, it turned out I did over 100 samples. I got mostly from autopsy, and I discovered that I used to
these were skin samples,
from
right?
Skin, the mesial aspect of the mid-upper arm, because there's no ultraviolet light there, and I used to do it bilaterally, and I would cryopreserve the explants, and I'd start fresh cultures, and every one of them would undergo clonal senescence. So it was the major confirmation of Hayflick. And I had evidence of a decline as a function of donor age, and that's still controversial. And my interpretation on that is that it has to do with the fact that they were autopsy, not biopsy material. We could talk more about why I think that's important. But at any rate, I did eventually 4 patients with Werner syndrome, and they fell 2 to 3 standard deviations below the means of age-matched controls. And they also had one patient with a Hutchinson-Gilford progeroid syndrome, and that ranked almost at the bottom of their group.
So this was the ability of cells to proliferate.
Right. We measured the cumulative population doublings for it. So that was impressive. I said, maybe this does have something to aging. And then we started to look at the phenotype, and Holger Hahn was an outstanding postdoc, became a faculty member, and then became chair of human genetics at the University of Würzburg. He's retired. He's one of 3 postdocs retired before I did, basically. So he showed that there was a variegated translocation mosaicism. There were all kinds of reciprocal translocation, deletions, inversions. So at the chromosomal level, it was a mutator phenotype. And this guy here with Ken Figuchi helped us show that it was a mutator phenotype at the molecular level. There were missense, a lot of large deletions as well we saw. And that was generalized. I did a kind of a review of, at that time, Victor Mikusik's Mendelian Inheritance of Man. We had Mendelian Inheritance of Man. We just had these volumes, right?
Yeah.
It wasn't online.
Yeah.
So I went page by page by page, and it had a bunch of tables of phenotypes that are like aging to see what falls out. And what fell out were a lot of syndromes that, for which now we have evidence is genomic instability. That seemed to be a big theme in that. That was gratifying as well. So anyway, so I stuck with that for a very long time.
So part of the research, at least at that point, was very heavily cytogenetics oriented, and I think it was either around or shortly before that time that the clinical cytogenetics service developed here, and you were instrumental in that process.
Yes.
Can you tell us a little bit about that as a a new clinical service, the first genome scale technology here, enabled by
It
chromosomes.
was 1963, I think. Dave Hogness, who was the dean of the medical school, he was my teacher in endocrinology as a medical student. Then he became president at the University of Washington.
John Hogness.
John, did I say Dave? He's a famous geneticist.
Right.
That's his brother, Dave. I got them mixed
Fly
up.
geneticist.
Yes, a fly
no less.
geneticist. But this was John. Thank you.
Uh-huh.
Unfortunately, John passed away a few years ago. So anyway, he gave me $10,000 to set up a diagnostic clinical cytogenetics lab in the hospital. And Phil Fialco was my clinical liaison. He was a fellow with Arno, medical geneticist. He became dean of the medical school later on, and we lost him in this terrible avalanche in the Himalayas. So we could diagnose Down syndrome, Turner syndrome, Klinefelter syndrome, and also the Philadelphia chromosome, which we diagnosed as a translocation. And I could remember later on, Janet Rowley showed it was a reciprocal translocation, I'm sorry, we diagnosed as a deletion. The long arm of chromosome 22, but it was a 922 translocation, according to Janet Rowley. I remember exams that I gave the medical students, multiple choice. If they wrote down a translocation, I took off points. I still feel guilty about that after all these years. So anyway, after 1 year, it was we thought it was quite successful. And the clinicians liked it, Phil liked it. And so I wrote a letter to the King County Medical Society asking if they would approve it for coverage by medical insurance. In those days, you had to get the approval of the King County Medical Society. So I got a letter which I wish I could have it saved. It said that they're terribly sorry that in their opinion, this is still a research procedure. Here we're diagnosing Down syndrome. So we closed the lab. We couldn't sustain it. Then when was it? About 1972, Arno had this wonderful program that involved a more what was it called, Arno? Again, the program where we had a piece of the consortium where we were doing growing amniotic fluid cells. There were a variety of individuals in different departments working on aspects of genetic for translational work. Yeah, it was kind of a center. Yeah.
Center for Inherited Disease.
Center for Inherited Disease. Yeah. Anyway, we had this focus on amniotic fluid, looking towards prenatal diagnosis, and we did actually a lot of starch cell electrophoresis, and we did clonal analysis and showed they were different from fibroblasts.
But this was all still research. You were not doing diagnostic cytogenetics.
So anyway, these clinicians came back to me begging to start a prenatal diagnosis lab, particularly the obstetricians. And I said no, to go through this again after spending the time. And finally, Holger Hearn was there. And God, it was a real hot shot in cytogenetics. So I said, you do it. So he did it. And if you go down the lab, you see a picture of Holger Hearn. And then this was great romance between Christine Desèche and
this part can be deleted.
Yeah, that came out later on. And that was fantastic because we recruited Christine, and now she's for many, many years been the co-director of the lab with Tom Norwood, and it's been an enormously successful lab. And out of that has come labs at Swedish Hospital, at Children's Hospital, and our chief technician made a lot of money by starting a private lab in town. It's still going on. So that was a quite successful venture, I think. Yeah. So anyway, that's the cytogenetic story.
Let me tell you one story.
Yeah, go ahead.
What
Yeah.
you said about cytogenetics.
How can we get you closer to a microphone? Can we do that? Yeah.
Well,
There you go. Yeah.
but I thought.
Here, we're getting a
Hold on a
microphone
second.
for you. Yeah. Here we
Should I
go.
get up?
Great.
No. Stay put.
No,
It's
stay
coming to
where
you.
you are. It's
It's coming to you.
Yeah. Okay.
I thought that since cytogenetics was something that should be available in general to doctors,
Yeah.
but I insisted pretty much that ours unit of medical genetics law, there should be a research unit. We would do clinical work, but the clinical work shouldn't take over the research.
Sure.
So in fact, we thought, let's give it to pathology. And George Stan, for years, told me, you see what you did to us? We could have had plenty minutes, lots of money from cytogenetics, if you gave it all away to pathology.
Well, Christine will tell you, we don't make that much money. So you could tell George that. Yeah. But it's wonderful, as an aside, what's happening that I see now, what's the wonderful interaction between genome sciences and the Department of Pathology. Bob Waterston was a keynote speaker at pathology at the retreat recently, and Debbie Nickerson is on the search committee for the new chair, and so that interaction is wonderful. I think we're going to see more of that in the future.
So George, who are the other people at that particular juncture? This was the 70s, 80s here, in genetics and medical genetics that you spent a lot of time with or spoke with as colleagues.
Yeah.
In terms of ideas or directions.
Yeah, well, of course, the key people were Arno and Herschel and Stan. But Ben Hall, we're getting a little ahead of the story. When I started the Alzheimer's Disease Research Center, I went to Ben Hall and asked him, could you recommend a hotshot or promising molecular geneticist? And he suggested Jerry Schellenberg, who was struggling in neurology, division of neurology at that time, barely making a living at working on calcium and metabolism. And he's now very famous. And that also, I went to Phil Fialco to try to recruit a statistical geneticist. We had Joe, but we didn't want to bug Joe about human stuff. We wanted to get somebody who do the statistical genetics for us. And we got Ellen Weissman from Stanford, and that was a tremendous coup. And then we had Tom Bird, who was a neurogeneticist, and had lots of pedigrees with autosomal dominantly inherited early onset familial disease. And I had a big pedigree that a pathologist sent me. He was part of that pedigree. So when the RFA came out for Alzheimer's Center, there was about that time, Jim Gusella from Mass General Hospital had mapped the Huntington gene to 4P, and I thought that was sensational. We could do linkage analysis with these pedigrees. Right. And it turned out that of the 3 autosomal dominant genes, our center mapped 2 of them, the presenilin 1 and presenilin 2, and Jerry Schellenberg, along with Rudy Tanzi from Mass General, they cloned presenilin 2. And the presenilin 2 is really an interesting story that, thanks to Tom Bird and his associates, it turns out that these pedigrees, only a small number were found compared to presenilin 1, but almost all of them are traced to one of 2 little villages on the west bank of the Volga River. They're called Volga Germans. Right. And it turns out that around 1760, Catherine the Great, who was herself from Germany, from the Hesse region around Frankfurt, she encouraged her countrymen to settle on this nice farmland on the west bank of the Volga River, and assured them they wouldn't have to serve in the Tsar's army, and they could keep their language and customs. And so some of them, they brought with them autosomal dominant gene, the presenilin 2 gene. And as a matter of fact, it's always possible that Alzheimer's first patient, who was around 51, 52, may have had a presenilin 2 mutation. We don't know that. But at any rate, Tom found that families in places like North Dakota, and his genetics associate would, over the telephone, would ask people about their origins, and she'd get answers like, "Well, it's a little village you'd never heard of it." She said, "Is it the village of Walter?" and be silence on the other end of the phone. the power of family history. So that was fun to see that develop.
So there were sort of two other things, I guess, coming to fruition at that point. I mean, one is the Alzheimer's Center started, and you were the first director of that center. The other part of this was the growing interest in molecular genetics, I guess, applied to human disease. And you mentioned the positional cloning of the presenilin genes, and also you were involved in the positional cloning of the Werner syndrome gene eventually.
That's right. Yeah, it was 1996.
Because this was a mystery for a long time, and there was a lot of speculation as to what that was.
Yeah.
So it must have been some measure of satisfaction to have finally identified this.
Yeah. There was a postdoc I had, John McKay, and he tried to push the lab into moving into more aggressively to see if we could map this gene first and then clone it. And I went to Ellen Weissman, and Ellen suggested homozygosity mapping.
Right.
And it worked. We mapped it to chromosome 8, and then with the help of Darwin, what is it
Darwin
called?
Molecular at that
Molecular
point,
in Bothell. That was one of Lee Hood's operations.
Right.
They had this sequencing, and Chang and Yu, who was on my training grant, I started a genetic approaches to aging training grant.
I was going to come back to that in a minute.
And Junko Oshima, who's still with me, and Fuki Hisami, who's here now, with the help of these people at Darwin, they nailed it. Actually, I remember the first author was Chang and Yu, and one of them was going telomeric and one was going centromeric, and Chang Yin won. He got the gene first. So that was really exciting because it turned out to be a member of the RECQ family of helicases, and it's an unusual member because it had, later on we found with Larry Loeb and Judy Campisi, it's had an exonuclease domain which separated out from the others. And then, well, you're now leading a program project on Werner syndrome, which is very gratifying.
There's still mysteries.
Yeah, still a lot, that we have to learn about it. Yeah. And actually, we did a lot of work in the Alzheimer's Center on trying to make mouse models. We made, with Ken Fuguchi, one of the first putative mouse models. Actually, we tried to, we overexpressed, and there was, Samir Deeb was very helpful in that regard. We overexpressed a domain of the amyloid precursor protein such that it would be secreted. And we're interested, and it had a promoter that was very promiscuous in many, many different tissues. And we had the notion, since the amyloid precursor protein was stored in large amounts in platelets, that if we could show that we can make the disease in the brain, and if we can now get rid of platelets by plasmapheresis, we might ameliorate it. It might have a clinical application. So it turned out that even though some of our colleagues who could measure A beta 1 to 40, A beta 1 to 42, the different moieties that are associated with the with Alzheimer's disease, peptides are about 40, 43 amino acids long, that we had huge amounts, the largest they'd ever seen in the plasma, and they couldn't get past into the brain in suitable concentration. We didn't get plaques, but we discovered a new disease, a model for a human disease, the commonest degenerative disease of skeletal muscle called inclusion body myositis. And it was a dead ringer for that. It had amyloid and tau and so on. So that was a surprise that came out of that work.
So I know when we were talking about amyloid and Alzheimer's disease, you were telling me this was one of the big fish that got away.
Oh,
do you want to talk a little bit about that?
This was 1985.
This was an opportunity that was missed.
George Glenner, 1980. George Glenner was the first guy to describe any amyloid. It was the IgG light chain or type of amyloid, and for many years they thought that was the only amyloid. Earl Bendit discovered the second amyloid. It was from high-density lipoproteins called amyloid AA, associated with chronic inflammatory diseases like rheumatoid arthritis, ulcerative colitis, and so on. And George Glenner got interested when he moved from the NIH down to California, UC San Diego. He got interested in the stuff that was in the brain in Alzheimer's disease. He got a Down syndrome patient when they have a lot in the P arachnoid in the blood vessels. So he managed to solubilize it with acid, and he got the N-terminal amino acid sequence of it. So I went to Samir Deeb. I said, Samir, he could clone anything.
Yeah.
I said, we've got the N-terminal amino acid sequence. Let's go after that puppy. And so someone who was very, very influential told me that, no, this is already being done.
Yeah.
So, okay, it's already being done. And so it turned out it took a long time. When I was in 1987, it was cloned by the Muller-Hill lab in Cologne with Colin Masters and those people. And I think it took Samir about 2 weeks or so to confirm it or so. It was very so that we lost a chance. The Alzheimer's disease could have been the first to get to clone the beta amyloid precursor protein. Poor George Glenner, incidentally, he died of amyloidosis, a different kind of amyloidosis. It was a transthyretin or retinoic acid binding protein type of amyloidosis. And the people in Portugal, I visited the center for doing all the sequencing for this type of amyloid, and they had George Glenner's DNA, and he had wild type. He didn't have a mutation. And it's apparently extraordinarily common, this type of transthyretin cardiac deposition of amyloid. In fact, people now are doing autopsies on supercentenarians, and almost everyone, there's just a handful of them, almost everyone has loads of transthyretin in their cardiac. So it's kind of a rate-limiting phenomenon, this type of beta fibrillosis, the beta pleated sheet type of proteins, either anti-parallel or parallel, that they aggregate and are highly insoluble. So
one of the things that came up before I wanted to cycle back to, you mentioned that at the same time the biology of aging research was starting, you got interested in applying for a training grant, the Genetic Approaches to Aging training grant, that I think the non-competing renewal, I can't remember what year, was just submitted, literally in
32, I think.
In the last day or two.
Yeah, so, right. Poor Peter, thank God.
So talk a little bit about the history of that and the relationship with the NIA, because I think that relationship for a long time was a very productive one, and that training grant has been remarkably successful.
It has, and John Gallant was one of the early participants in it, with his interest in translational infidelity and the Orgel hypothesis.
As a mentor, not as a subject.
As a mentor, right. But he had students who he supported in those days. And actually, we'd love to have even more people from genome sciences, because it's called Genetic Approaches to Aging Research.
Right.
Anyway, it's been wonderful, and it was the first of that type. And it's still going on. And Peter Rabinowitz, who was my graduate student, and from genetics, he was a genetic graduate student, he not only is keeping that going, to my old program project going, and he got, on his own initiative, he got the first what's called a Nathan Schock Center for Excellence in the Basic Biology of Aging. And I teased my colleagues in San Antonio, who got the second one, that we were first. Turns out they got theirs about 10 seconds after voted in, but we are the first. Nathan,
you're not going to let them forget it.
Right, we're not going to forget that. Yeah.
So another, I don't know if it was around this time that you made the connection with the Ellison Foundation. They've got a biology of aging program that funds, I think, especially junior investigators who are exceptionally promising. And can you talk a little bit about how that developed? And you still have a continuing role in the Ellison Foundation, don't you?
It's my last year. Well, one of the sabbaticals, we didn't talk about my wonderful sabbaticals. That's really one thing I would recommend for young people.
That's the advice
people are giving it up.
We'll
come to that. Okay. Anyway, that was a sabbatical I had with Josh Lederberg at the Rockefeller, just a mini sabbatical. And he asked me if I could join a new initiative. He was the first one I asked
So
on the
when was
biology
this,
of
George?
aging.
This was
It
on
was 15
sabbatical.
years ago.
15 years ago.
15 years ago. He had met Larry Ellison, who's the CEO of Oracle, and he was the inventor of the first relational database, which has now made him rich and famous. Josh got him interested in doing something with his money and thought that the thing that was beginning to pay off, and it was right, largely because of what was going on with Caenorhabditis elegans, to try to fund a large project on basic research in aging. So it started the Ellison Medical Foundation. So I was on the first scientific advisory board, and Josh was chair, and I'm now chair after he passed away. I'm rotating off along with Eric Kandel, the famous Nobel laureate neurobiologist, and Jerry Weissman, who's the editor-in-chief of the FASB Journal, kind of a polymath, and Dick Sprott, who's the first executive director. We're rotating off on 1 September to get some new blood and different people with different ideas. But it's been enormously successful. We have a series of new scholar applicants, and there can be now, we've asked the individual institutions to nominate up to 2 individuals for this award. And we've had several from this campus. And we have senior scholar awards, and there we typically have gotten over the years of about 200 letters of intent for these awards. We first gave 10, now we give 25. And just since this crisis at the NIH, in 1 year it went up from 200 to 300 letters of intent. So as a piece of evidence for how much pain there is out there in trying to keep funding, even for senior people, very good senior people. So anyway, it's now accounts for about a quarter of the budget, equivalent of a quarter of the budget of the Division of Basic Biology at the National Institute of Aging. Which is relatively small compared to what's going on for Alzheimer's disease, which hovers around 50% of the total budget.
I mean, another aspect of this, I'm reminded, it was about 10 years ago that we did this symposium on the biology of aging here. And this was in part in your honor. And, we had a stellar cast that came literally from around the world to present science at that meeting. But I was struck equally by a comment every single one of those speakers made, George, which was that they valued you as a mentor, as a friend, and as a confidant or a consultant who gave them, critical pieces of advice or supported them when they were often junior faculty members. And I've come to know more and more of this. I've certainly benefited from the mentorship aspect of this, and I was wondering where this came from, because you seem to have an extraordinary breadth of mentees in a sense, and you've been very successful at encouraging a lot of young people, not only in the biology of aging, but in their careers.
Yeah.
So how did that develop?
Thanks for that. I just I think that's part of my that physician instinct, to try to help people. Yeah. And I still
do no harm.
I still feel that way.
Yeah.
Especially young people, and it's so difficult right now. At that, who the star of that Festschrift was my daughter Kelsey.
Right.
She gave the best talk, I think, of anyone there. It blew me away. She has a hot paper and press for Cell right now.
Well, she'd been under your tutelage longer than the other individuals there, George.
For a long time. Since the little girl who came from Scotland with a wonderful accent. Yeah. So we wanted to leave a little bit of time to talk about advice and to talk about the future, and I guess let's start with advice. I mean, other than taking sabbaticals every opportunity you have. There we go. Yeah.
And I'm thinking especially about advice to someone who might be a junior faculty member or a student or a postdoc.
How about a senior faculty member?
Well, we can come to that as well.
Well, I can tell you what my advice is, and I'm doing it right now.
Yeah.
I'm looking for a fabulously wealthy, moderately attractive widow who could fund my lab. that would be, because I'm having the same stress. It's kind of a controlled atrophy is going on with my lab. That's the outstanding piece of advice.
Okay, I'll take that under advisement.
The other thing, I would say that I think it's great to have a consistent theme and to go deeper and deeper layers of depth if it's working over a whole career. However, I think it'd make it more fun if there were 10% or so of the budget that you had. You set it aside for taking flyers on completely wild things, because it's fun and something really interesting and maybe transformative could come out of that. I think that would be my advice for young people. I think that it's interesting. The MDs tend to be too goal-oriented, and the PhDs are a little laid back, a little hang loose. I think hanging loose is good, to pick up some low-hanging fruits in a different area and go after it. I think that's something you should. It's all right to have that focus, but not to be obsessive-compulsive about it, and keep your eyeballs open for other opportunities that come up would be my advice.
I guess I had forgotten a piece of history here. You founded the MD PhD program at the University of Washington.
I did.
You not?
I think I'm more proud of that than almost anything else. And thanks to Larry Loeb, has shepherded it for more years than I did. And now Marshall Hurwitz is doing a beautiful job. And that's so important. And with some fantastic students that have come out of that. So I'm quite proud of that. That was, I think I wrote the grant for that, well, I can't even remember how many years ago it was, maybe 1969, something like that. Yeah. So the future, I would say, I'm so proud of this department. It's a fantastic department. And Jay Schender is sitting there, and Debbie Nickerson.
No,
not Jay.
That's Raghu.
Raghu, sorry, not Jay. Raghu.
But I can be Jay for a day.
You can be Jay for a day, yeah, Raghu. So anyway, and Debbie particularly has been very helpful, and Gail Jarvik for what we're struggling to do with Werner syndrome. We have an international registry, so we get cases from all over the world, and docs say it's Werner's, and it's often a little bit funny about the clinical presentation. And so with Junko Oshima, we sequence all the coding region. We do Westerns. The protein is normal, the amount of normal, the migration is normal, so something else. So we had about 48 cases like that. And we went at candidate genes, and the first candidate that we looked at was lamin AC, and there's a mutation in Hutchinson-Gilford progeria syndrome. It's on the C-terminal domain. They have a 50 amino acid deletion so that they lose the site for clipping off the post-translational farnesylation. They drag this stuff into the membrane. But our guys, they're more N-terminal, and they're different ones. They're having different protein-protein interactions. So that was gratifying. But now we have 41 cases. We don't know what it is. And with this wonderful exome sequencing, we're hoping with chipping and an exome sequence, we're trying desperately to get nuclear pedigrees. And we might nail some new loci. They're out there. It's compelling that this is a genetic situation. So we're excited about that kind of interaction as well. And the future, the other thing is, I noticed this copy of Cell here when I came in. This is my own copy. I brought it along. And I participate in the Medical Genetics Journal Club, as Arno and Stan have done for so many years. And I'm thinking of presenting a paper here because it illustrates what I think the future is in medical genetics, is to spend more time on looking at situations where you have a genetic factor, but and you have variable penetrance, but there's something in the environment that makes the difference. And here, they're looking at congenital scoliosis. This is a group mainly from Australia. And they show that, there's some mutations in the Notch pathway, and that the penetrance is first of all, they find that in human families, and it's the penetrance is variable depending on what mutation you find. But now they make a mouse model, and they find that if you produce a moderate to mild hypoxic challenge during embryogenesis, not enough oxygen, then the penetrance goes way up. So it's a model for a gene environment interaction and for
during
public health.
During development.
Yeah, during development. Yeah, during development, and it's a very important issue in public health, and I think we need to do a lot more of that. Just one other example that comes up is in Crohn's disease. Crohn's disease. There was a paper in 2010, also in Cell, where that also there are about 20 different loci, and there's a risk factor, but they don't know what goes with the genetic susceptibility. And in this paper, of all things, there's a viral agent, a specific viral agent that interacts with the genetic susceptibility and produces the inflammation. I think that that's a really important thing to pursue, because the environment you can do something about. In principle, if you know what it is, and there are probably lots of environmental agents that are making a difference in somebody who has susceptibility for that.
So environmental modifiers. Genetic risk here.
And of course the gene actor, and then personalized medicine also. You're going to deal with so many modifiers and environmental interactions. It could take a long time to really interpret all that. The $1,000 genome sequence, what is it, $1 million to really fully understand all the modifiers. I believe that.
So I think we're close to time here, George. Are you willing to take a few questions?
Sure, love to.
All right.
If you find a rich widow, would you please make her introduction?
I can't solve that problem. I just wonder, you'd have to say, I think, that in working on the senescence of cell lineages, that you made a good bet back then. I mean, this is still, last I heard, it was still important in aging. Was that blind luck, or do you think somehow you sensed something about the potential importance of this?
Well, that's part of the message. It was, you kind of follow your nose. I got interested in vanadium, and vanadium brought me into aspergillus, and aspergillus brought me into cell culture, and cell culture brought me into aging. It just it happens that way. There's a lot of stochastic processes that are going on, well, it's the story is quite interesting now in terms of telomere biology, right? And that's a big deal right now. And the stem cells, of course, have telomerase, and the germline has telomerase, but these other cell types, somatic cell types, don't. So that seems to be a major. But there are other reasons for getting out of the cycle. Incidentally, since Joe was asked the question, I was chatting when he came early. I was chatting with him about my naivete in evolutionary biology, and that's another thing I would love to see more of in the department, because we're having it now. It's Maitreya's, Dunham involved. I'd like to see even more of that, because it's the most compelling aspect of biology. And anyway, I remember giving a talk in the genetics department on a quite young man. And I was really excited because I had shown that the replicative potential, the number of clones that I could get out from the aorta of a long-lived mouse, Paramyscus leucopus, 8 years, versus an ordinary lab mouse, lives 4 years, it was dramatically different. The long-lived guys had a lot of cells, and they all went down with age, but the Paramyscus had a lot. And I said, gee, this is interesting, because these are closely related species. And Joe got up, I think he said, it's my responsibility here to point out that these closely related species differ evolutionarily by about 30 million years. So I kind of shrunk down a little bit. And he's been helping me ever since. Teach me a little. principles of evolutionary biology.
We got other questions?
Yeah.
Here?
Yeah, Bob?
Yeah, I have one. I mean, you've watched the medical center change from a relatively small
Yes.
place to a massive institution. And I wonder what your take on that is and how it changes the research environment.
Yeah, well, it's still, Bob, it's a good question, and it's true that you don't get to know a large percentage of people because it's so big, but I'm impressed that this is a wonderful place to be because it's kind of an open door policy. Postdocs and graduate students can go, in my experience, anywhere to learn a new technique. It's very different than a lot of other institutions I know about. So that's a good sign, and I think we're doing very well despite this grand crisis. I think we're doing extraordinarily well. Although I think in our department of pathology, what is it? The state provides 2.3% of our budget or something like that. I mean, we might as well be a private institution. And thank God we're still competitive, but it's worrisome. So I think there's opportunity as it gets bigger and bigger for more collaborative ventures and just more expertise in diverse areas. This is a terrific medical school, and it's interesting that it's also very strong in primary care medicine, family medicine. It's very strong in a lot of other areas. In my late chairman, Nelson Fausto, who unfortunately passed away recently, multiple myeloma. I visited him when he was in the rehab medicine because he had some spinal surgery. So I had to warn him. I said, Nelson, you're in a second-rate operation here. Turned out they were voted second in the country. Yeah. Sometimes they're voted third, but I felt it was my duty to warn him about that. So we've done very well in many areas.
Charles, do you have a question?
Yeah, I too have been impressed with the MD PhD program.
Yeah.
How do you see that developing? Of course, I'm not asking you to give Marshall Horowitz's marching orders, but if you look at these changes in medicine and funding in the PhD, what do you think the route might be to accommodate those?
Oh boy,
And still maintain a few investigators.
Yeah, to keep them going. Well, they're usually so competitive, and if they have, I've never felt that we lose them if they go into a basic science thing, because they could pick up a telephone, and if they have an idea that's clinically relevant, they understand it, and they can talk to a clinician to get studies. So I think that's fine if they go to basic science departments, but they're more likely to make a living if they're doing some clinical work in a clinical department. And I think my advice generally is to go to a major medical center where you do have an option to be not only a specialist, but a subspecialist. And for example, those are the people who thrive. Like Peter Byers, for example. Peter Byers sees patients who have connective tissue disorders, mostly osteogenesis imperfecta, Marfan syndrome, Ehlers-Danlos syndrome. And so he biopsies the patient, he discovers new mutation. It's one world. It's all together, you see. Whereas some of the kids who went into, for example, hematology, oncology, and they get to transplant patients who are terribly sick, they're up all night, and they have an NIH grant on some basic problem. It's really a tough life, and I think what suffers is their family. In fact, some of the kids that get into trouble, it's usually emotional kind of problems they have. I think that another example works if you are in a big place and you could see, let's say, a pediatrician sees only kids with type 1 diabetes, and they do their research in type 1 diabetes. But if you have to do ward rounds and deal with all comers, that's very difficult because you can't make mistakes. You can't miss a meningitis if you're on call. So how to do that? You have to have the luxury of being at a really major medical center in order to pull that off. Yeah.
Well, I think we're at time, or perhaps slightly over. So I think thanks again, George.
Okay,
this
good.
was great, and
Thanks for everybody
enjoy
coming. Yeah, thanks for
enjoy
coming.
some wine and.